Well, next we have another session on research and drug development updates from our industry partners. We're going to begin with Dr. Jahannaz Dastgir from Regenxbio. Dr. Dastgir is the Senior Medical Director and Clinical Development Lead of the RGX-202 program focused on AAV8 gene therapy for Duchenne muscular dystrophy. She completed her pediatric residency at the University of Connecticut's Children's Hospital, her child neurology training at Boston Children's Hospital. She currently maintains a once-weekly neuromuscular clinic in New Jersey, continuing to care for children and young adults with childhood onset neuromuscular disorders. And we will just have one speaker followed by questions. We have 3 speakers in this segment, and we begin with Dr. Dastgir. Welcome.
Hey, thank you. Can you guys all hear me okay? All right, thank you so much to Defeat Duchenne for having us here today. I really appreciate it, and I apologize if my voice is a little scratchy. I've been working on it. So just to give you, we just wanted to give you an update on RGX-202. So this is Regenxbio's investigational gene therapy for the treatment of Duchenne muscular dystrophy. How do I switch the slides up here? I apologize. Is it on here? Thank you. I didn't check that before I got started. Okay, so just as you've seen some of the other companies provide, we have forward-looking statements from Regenx here that are a lot of words, but kind of we have to put it up here. So I'll switch on to the next part of the content. So Regenxbio is a company that is seeking to improve the lives of improve lives through the curative potential of gene therapy. We are a leader in AAV gene therapy. We have exclusive rights to the NAV technology platform. We have a broad internal pipeline across multiple therapeutic areas, including neuromuscular disorders. We have end-to-end capabilities from research through clinical development and commercial-ready manufacturing. And we have proprietary AAV manufacturing, including a NAV Express platform. And just to be clear, AAV is adeno-associated virus that's typically used for gene therapy. So RGX-202 is an optimized microdystrophin gene therapy with unique features. We are partnering with the Duchenne community to guide development. So being a part of a community like this is so important to us. We always encourage your feedback, any ideas you have. We have preclinical studies completed providing evidence of potential benefit. We have a US FDA IND clearance to evaluate our drug, RGX-202, in a phase 1/2 trial that's named Affinity Duchenne. And we plan to initiate dosing in the first half of next year, 2023. So RGX-202 is a microdystrophin designed to retain the key elements of full-length dystrophin, including the C-terminal domain. So dystrophin by itself is a gene that's very long. We all know it has about 79 exons in it. And it's kind of hard to package that in the AAV or adeno-associated virus vector. So what happens is a lot of the companies that you'll be hearing from that do create gene therapy have their own constructed version of the microdystrophin. So what differs between ours and others is that we have included one part of it called the C-terminal domain. And the reason why we did that is because it kind of links to some other key proteins in the muscle cell membrane. And we've had preclinical work on animal models, including the MDX mouse, which has showed an increased resistance to muscle damage by including that C-terminal domain. And the way that gene therapy is delivered is that we have the AAV8 vector. So you'll hear from other companies as well. They're using different adeno-associated viruses. So we use AAV8, and we also have a promoter that is used to construct this microdystrophin gene. So the promoter is what kind of drives the microdystrophin to the target. So we use SPC512, and then we have the microdystrophin gene there. And just going through the slide here, the vector delivers the transgene to the skeletal and heart muscles in the body. The promoter drives the expression of the protein in muscles. And then the microdystrophin gene encodes the novel protein that we were talking about with the unique C-terminal domain. And this is given as a one-time IV infusion. That's it. There's a lot that leads up to that, but the dosing of the drug is a single dose. So RGX-202 has preclinical evidence in the Duchenne muscular dystrophy animal model, also known as the MDX mouse. So here you can see data from the mice. So the first, I don't have a pointer, but it's okay. I'll go through the slides here. So where you can see the title that it says that there's robust microdystrophin expression in skeletal and cardiac muscle, you can see that there are varying doses, and the doses are escalating of our RGX-202. And as the dose goes up, we see increased expression of microdystrophin in the skeletal and cardiac muscle. So what does that mean? So back in the day, kind of even when I was started training, a lot of the Duchenne muscular dystrophy diagnoses were made based on muscle biopsy and dystrophin staining. So this is kind of checking for that microdystrophin level, not dystrophin, because that's not what we're giving to patients. We're giving microdystrophin. So this is the microdystrophin levels that we're assessing for. And then we can also see a significant increase in muscle function and strength that correlates with the increasing doses of drug. And this is in mouse muscle function and strength testing. At the bottom, you can see improvement in muscle fibrosis. So if you look where it says wild type, this is a mouse that has no expression of Duchenne muscular dystrophy. So it's a wild type non-MDX mouse. And the subsequent ones are MDX mice. And if you see the first picture there, that is an MDX mouse who has not received any drug at all. And you see a lot of fibrosis. Fibrosis is typically scar tissue or fatty replacement of the muscle that happens over time as your muscle cells may degenerate and regenerate. With the lower dose at dose 1, you can see that fibrosis has improved. And as you go on to the higher dose at dose 4, it's gotten much better. So our affinity trial, we have preparation for initiation right now. And again, as I mentioned before, we're going to be starting in the first half of 2023. And the steps are as follows. So we have optimized RGX-202 with our AAV8 vector and SPC512 muscle-specific promoter. I'm in the unique C-terminal domain. And we have completed our preclinical IND-enabling studies, activities, which include the preclinical studies, manufacturing process development, key opinion leader, and very important community input. And we have a clinical study design in place. And where we are right now is getting ready, getting teed up. It's a very long process to get started, but we're going to get our clinical study up and running. We have US FDA clearance to proceed, and we're preparing for trial initiation, including readying clinical trial sites and manufacturing as well. So our study, so this is a little bit of an overview of our study. This is an open label, multi-center study, so many sites. There's no placebo here, to evaluate the safety, tolerability, and efficacy of a one-time dose of RGX-202 in boys with Duchenne muscular dystrophy. So the boys will be 4 to 11 years of age. They do have to be ambulatory, so they have to be able to complete some baseline timed function tests. We are looking for specific mutations within the dystrophin gene. In order to receive any gene therapy, you're typically tested for AAV antibodies to the specific vector you're using, because the drug would be basically ineffective if you had antibodies. So we will be checking for antibodies to the AAV8 capsid, and additional criteria will apply. And then the key outcome measures are going to be a safety assessment. We're going to check for microdystrophin protein levels in the muscle as measured by biopsy. We're going to be checking skeletal and heart muscle as measured by MRI. And we're going to be doing strength and function measurements, such as the North Star Ambulatory Assessment, or NSAA, and timed function tests. So in summary, RGX-202 is an optimized microdystrophin gene with unique features. We are very eager and excited to partner with the Duchenne community to guide development. We have preclinical studies completed, providing evidence for potential benefit. We have US FDA IND clearance to evaluate RGX-202 in our affinity Duchenne trial, and we plan to initiate dosing in the first half of 2023. That's it. Thank you so much for your time.
Great.
Thank you, Naz.
Yeah.
And Craig Campbell, Dr. Campbell has agreed to moderate this segment of questions and presentation. So,
Yeah.
Dr. Campbell.
Yeah, questions for anyone? Anything coming through? So Dr. Dastgir, thanks for that
Sure.
great presentation. Tell me, I mean, as you know, there's been a number of learnings from current gene therapy trials. How have those things, about reaction to the viral vector, some of the immune reactions we're seeing to the dystrophin product, how has that informed your preclinical development of this, of your particular vector and dystrophin cassette?
Thank you for bringing up that question. So this is an important area for us. We have a team that really is dedicated to kind of addressing this issue. We are very concerned about the safety of any patient enrolled in our trial. We have used a lot of the guidance and feedback to kind of, I'm sure you're familiar with the consortium that was developed through some of the companies that are already dosing patients, and like kind of picking up on which patients with certain mutations might have a worse reaction. We have taken that heavily into consideration in the mutations that we will be included in our trial. So that was one way. And then the other thing is also being very careful to select certain immunosuppression drugs that we will be using prior to dosing, and the duration after dosing. We have designed to make sure that we are addressing this, like kind of hitting it head on as carefully and with the safety of the patient in mind.
Yeah, and I don't know if everyone will be aware, but for the families and the community, the DMD community, what Dr. Dastgir is referring to is that the companies who are developing therapies or in clinical trials for dystrophin gene replacement therapy have gotten together, which I think is admirable and maybe really a first, gotten together to kind of share knowledge and experience around some of the particular immune reactions that we're seeing in the dystrophin trial, and I think this has been a very important step for sorting through some of the concerning reactions that some of the boys have had in the trial. So good to hear you're part of that work. Yeah.
Thank you for the talk.
Sure.
I'm curious if you're evaluating, perhaps in the background, any strategies to cleanse the neutralizing antibodies in patients prior to receiving AAV therapy?
So we are not currently actively doing anything to address that, but that is definitely on our mind. It's something that I am very interested in personally, because I do think that as we have more and more studies going on and more people out in the community who are getting dosed, we're going to probably see a lot of people coming up positive for antibodies, which will render our future drugs ineffective in some ways unless we come up with an immunosuppression regimen that'll be effective to kind of suppress the immune response prior to dosing. So definitely something we think about and are very cautious to address.
Any online questions? No. Okay.
Okay.
Great. Thanks.
Great, thank you.
All right. Thank you so much.
Thank you, Dr. Dastgir. We have one more presenter in the area of treatments, and delighted to be able to introduce Dr. Ashish Dugar from Dyne Therapeutics. Dr. Dugar has deep experience across clinical, commercial, and patient advocacy, real-world evidence, and market access with leading drug development companies, and has proven success in building a global medical affairs organization in rare muscle disease. Dr. Dugar began his career at the National Institutes of Health. Dr. Dugar earned an MBA from the Pennsylvania State University and a PhD in pharmacology from the Pennsylvania State University College of Medicine. So it's my pleasure now to introduce from Dyne Therapeutics, Dr. Ashish Dugar.
Thank you, Rick. I'm just pulling up my slides here. Hopefully you can hear me okay. Great. Do you see my slide? Hopefully you all do. Great. So thank you very much, Rick. I'm Ash Dugar, the Global Head of Medical Affairs at Dyne Therapeutics. And first, I want to thank Rick, Nicola, and the entire Defeat Duchenne organization for inviting us to this meeting today to update the Duchenne community on our progress of our Duchenne program, specifically Dyne 251, which is our clinical candidate designed to treat patients with Duchenne who are amenable to exon 51 skipping. And following those two fantastic talks on gene therapy, we're going to switch gears a little bit to talk about exon skipping. These are our forward-looking statements. Please refer to our website if you're bored and would like more details on this. At Dyne, it all starts with our mission. We're laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with Duchenne being a priority. Our mission is core to who we are and what we do every single day. And I'm incredibly excited to inform you that we've started dosing participants in our Dyne 251 program. And our focus today is on sharing an update on our clinical trial progress and details about our clinical trial with Dyne 251. Over the last few years, we built a robust portfolio focused on rare muscle diseases. Myotonic dystrophy type 1 or DM1 and FSHD represent opportunities to develop first-in-class therapies. And in Duchenne, we're building a global franchise starting with our exon 51 program, and we're following this with programs for exons 53 and 45 and 44. This slide provides an overview of Duchenne, which you all know very well. Our goal is to develop a best-in-class exon skipping treatment targeted to skeletal, respiratory, and cardiac muscle that greatly increases dystrophin expression and has much less frequent dosing than current agents, with the hope of potentially stopping or reversing disease progression. So here I'll take a couple of minutes to describe the FORCE platform. This scientific platform is the basis for all of our neuromuscular therapeutic candidates. This platform drives our efforts to develop and deliver muscle-targeted therapeutics. It's expressly designed to address one major issue in muscle diseases, which is efficiently and reliably delivering antisense oligonucleotides, like PMOs, that target the genetic basis of diseases to skeletal, cardiac, and smooth muscle. As you can see, Dyne's FORCE platform consists of three parts: a proprietary antigen-binding fragment or an antibody fragment, or what we call a Fab, to enable targeted delivery of the antisense oligo or ASO to muscle, a clinically validated linker, and an ASO selected to target the genetic basis of disease. On the left side of the slide, you can see a Y-shaped structure, which is a monoclonal antibody. Well, we chose the top portion of this, our Fab, as I mentioned, because we believe it offers powerful advantages over full, larger monoclonal antibodies, including its smaller size, leading to better muscle penetration, lower overall protein load, and the potential for enhanced tolerability. The goal is ultimately to enable targeted, effective, and efficient delivery of our therapeutic candidates to skeletal, cardiac, and smooth muscle. On the bottom part of the slide, you can see the linker. We selected something called the Val-Cit linker for our platform based on its stability and clinically validated safety and efficacy in approved products. We believe this further de-risks our overall FORCE platform. Now we're utilizing the same Fab and linker across all of our neuromuscular programs, including Duchenne. The versatility and modularity of our scientific platform provides flexibility to deploy different types of ASOs that are rationally selected to match the target biology for each disease. Where dystrophin restoration is the key, as is the case in Duchenne, it's critical that we select an exon skipping phosphorodiamidate morpholino oligomer, or a PMO, that targets the specific mutation in the DMD gene. So in our current study, we're using a PMO that allows for skipping exon 51. Now, the mechanism of FORCE is different from other approaches. We are essentially using the cell's natural biology to deliver our therapeutic to muscle cells. Our Fab is engineered to bind tightly to something called the transferrin receptor 1, or TFR1. Now, the TFR1 is highly expressed across the surface of muscle cells, including skeletal, respiratory, cardiac, and smooth muscles. Now, the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our PMO to enter muscle cells and target the nucleus of those cells so that we can address the genetic basis of Duchenne. So as you can see, this is very different from an unconjugated PMO, which is injected into the body. And this is very different from gene therapy. There's no viral vector, there's no capsid, and we are striving to create a shortened form of dystrophin, not a engineered microdystrophin. So in short, we're able to harness what already exists on the surface of muscle cells to deliver our therapeutic to muscle with this receptor-mediated mechanism. By targeting muscle specifically using TFR1, we have the potential for Y therapeutic index, resulting in less frequent dosing than currently available agents and chronic administration. On this slide, we show our fourth platform in the MDX mouse model, which you've heard in previous talks, a standard animal model in Duchenne. These MDX mice have a mutation in exon 23. You can see force resulted in robust dystrophin production across key muscles affected by Duchenne with just a single dose of 30 milligrams per kilogram, and we evaluated the mice 4 weeks later. The top half of this slide shows some of our data demonstrating robust levels of dystrophin expression across key muscles. We were able to achieve high levels of dystrophin expression, about 50% in the quadriceps, 90% in the diaphragm, and nearly 80% in the heart. We were really excited to see these levels of dystrophin restoration, especially in the heart and diaphragm, where current PMOs have had limitations. On the bottom half of the slide, we show dystrophin localization in the sarcolemma muscle membrane after just that single dose. Now, this localization is important to see to provide confidence in potentially seeing functional benefit. And we saw profound results, including about 70% dystrophin positive fibers in the quadriceps and about 80% in the diaphragm and heart. Importantly, our fourth platform has shown to have durability of effect. This slide shows our data on dystrophin positive fibers in the quadriceps out to 12 weeks. So even out to 8 to 12 weeks, we saw sustained localization of dystrophin to the sarcolemma and the quadriceps, and we've also seen this in other muscles. Now, this is really important as we think about the frequency of dosing in our clinical trial. Next, we evaluated Dyne-251, our clinical candidate, in non-human primates or NHPs or monkeys. These studies are a key step in generating preclinical data before going into clinical trials. We measured exon skipping in the quadriceps, heart, and diaphragm. Please note that in monkeys, we cannot look at dystrophin restoration because these are healthy animals. The key message here is that with our fourth platform, time on therapy matters. Over a 2-month period, we observed robust exon 51 skipping across muscles. After 5 weekly 30 milligram per kilogram doses, Dyne-251 demonstrated about 50% exon skipping in the diaphragm, about 40% skipping in the heart, and about 20% exon skipping in the quadriceps. We also evaluated Dyne-251 in 5 and 13-week toxicity studies in monkeys. Overall, we saw that Dyne-251 had a favorable safety profile with no dose-limiting toxicities observed after weekly doses up to a maximally feasible dose, meaning we dosed monkeys at doses much higher than 30 milligrams per kilogram and saw a favorable safety profile. We're really pleased with this as we advanced into the clinic. Now, prior to starting our Dyne-251 clinical development program, we proactively obtained expert feedback from key opinion leaders such as Dr. Campbell. And we engaged various disciplines, including pediatric and adult neurology, cardiology, physical medicine and rehabilitation, pulmonology, and physical therapy. Importantly, we have gathered this feedback from across the globe. KOLs both in the US and outside of the US with experience running early and late stage trials have provided advice on our overall clinical development program, including our clinical trial design, our patient population, biomarker and functional endpoints, and key safety considerations. Importantly, they have helped us learn from natural history studies. Now, it's clear that key opinion leaders believe dystrophin continues to be a key endpoint for this condition. We've also received advice directly from patients and caregivers, as well as global advocacy leaders and the Global Duchenne Community Advisory Board, comprising of advocacy leaders from several countries around the world. They provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. This input has been critical in ensuring a patient-centric design and has included, but it's not limited to, key considerations for choosing a clinical trial, review of and input to our informed consent form, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. Based on this extensive feedback, we've developed this clinical study for Dyne 251 in patients amenable to exon 51 skipping. Our clinical trial for Dyne 251 is a global, multiple ascending dose, or MAD, placebo-controlled study with a long-term extension. It's important to note that following the placebo-controlled portion of the study, all patients will be able to receive active treatment. We are currently enrolling patients who have mutations amenable to exon 51 skipping. Our goal is to enroll approximately 46 male participants ages 4 to 16 years who are either ambulant or non-ambulant. As was pointed out earlier, we have been striving to listen to the community, and we understand that the non-ambulant population has been underserved for a long time. We plan to evaluate the following outcomes in the DELIVER trial: safety and tolerability, change from baseline in dystrophin protein levels by Western blot, muscle function relevant to both ambulatory and non-ambulatory participants. So that would include assessments to help us understand changes in lower and upper limb function, as well as lung and heart function. So we would be looking at measures like the NSAA, time function test, cardiac MRI, and FVC % predicted. And importantly, we will be dosing monthly and striving to dose even less frequently in the clinic. With the help of our expert advisors, we are very confident that our study design is robust and will measure key outcomes and is striving to manage the burden on patients. As I mentioned, this global clinical trial is currently recruiting. We are in the process of activating sites around the world, including Canada, and we are pleased to say that we do expect safety, tolerability, and dystrophin data in the second half of 2023. And finally, we have tried to take every opportunity to listen to the Duchenne community on their needs for next generation treatments. We're working urgently to deliver for the community, including families and healthcare providers, to bring a potentially transformative treatment forward. Thank you to the families for being generous with your thoughts, advice, and for the sacrifices you continue to make. And on behalf of Dyne, I would like to thank you all for your time and attention to listen to this presentation. And I'm pleased to take any questions. Thank you.
Dr. Dugar, thanks so much. And I'll turn it over to Dr. Campbell to manage the Q&A here. Thank you.
Okay.
Thanks, Ash, for being here remotely. And we saw Molly here yesterday, had a chance to chat with her. So any questions from the audience around? So again, this would fit yesterday, if you remember back to our talk, into those genetic-based therapies and a lot of activity in the exon skipping category, including Dyne coming with their product. I guess one of the, do you have something? Or?
We just have one question
great,
online right
great.
now.
Yeah, let's go.
Will other mutations be targeted in the future? And if this study is successful?
Yeah, great question. So absolutely, we are in the process of moving forward candidates for other mutations, such as those that are amenable to 53 skipping and 45 skipping. So we continue to build out this, this pipeline within Duchenne. And maybe I'll just point out one thing. So in that construct, in this FORCE platform, I think I failed to mention that the Fab, so the antigen binding fragment that targets the TFR receptor, which is found across muscle cells, and the linker, the thing that attaches the antisense to the Fab, they remain constant for all of our therapeutic candidates. So it is the PMO that we interchange. So this makes for more efficient non-clinical as well as clinical development. So we're able to actually continue to move forward, learn from one program to another in terms of efficacy and safety, so we can efficiently go through and develop candidates for other mutations.
Yeah, thanks. So just that's, I hope, hopefully clear to everyone. The challenges with exon skipping, as Ash has said, is, are twofold. One is that actual genetic trickery that we talked about yesterday of in the dystrophin gene of restoring the reading frame, but the other is getting the actual antisense into the muscle. So those are the two, and many companies are working to find the best way to get that antisense into the muscle, and this is a unique, I think there's another question online there.
Will you naturally proceed from the most common mutations to the less frequent ones?
Yeah, it's a question that we get as well as many other companies frequently. So currently our progression is moving from 51 skipping to 53 and 45 skipping, which are the next most prevalent mutations. So that is our current plan. But as I mentioned, we're trying to move as many of our candidates as forward as quickly as possible. And, it is certainly well understood that outside of the US, there really are no disease-modifying therapies available except for a select couple of other countries. And so we want to be able to serve as broad a population as possible. So we are starting with exon 51. And then I think the other, honestly, the other part of it is that there is a precedent, there is benchmark. I mean, there is a benchmark. Our goal is not to have something that's incremental to the current 51 treatment that's out there, at least approved in the US, but to bring something that is, transformative to the table. And so we really hope that our approach using the body's natural cell biology to drive the PMO into the cell, certainly shows what we have been seeing in the preclinical models.
Thanks. Rick.
Dr. Dugar, thank you. The question I have is, and I understand it'll be difficult to be very precise probably in your answer, but if you are targeting 51 first, let's say that's successful, let's just move down the road a little bit and so on and so forth. What kind of timelines then to address the other deletions? Are we looking at months, years, decade?
Yeah.
What do you see? And I realize it's probably an unfair question, but your best assessment of where we might be if this 51 is successful.
Sure. No, thank you. It's a tough question, but it's actually a very fair question. It's been, it's really unfortunate that therapies haven't been approved outside of the US in terms of exon skipping therapy. So I think it's a very fair question. I don't have a good answer for you. What I can tell you is it will be important for us to be able to generate some data from our 51 skipping program as we continue to advance our candidates for other exons. We need to learn somewhat from our current program. So the good thing is we are in the clinic, we are moving forward, we are opening and activating sites for the 51 program, and we'll continue to learn as we generate data in the 51 program. But it doesn't mean that it's sequential in nature. There's a very clear non-clinical path that we have to go down, generate some data in animals. So we're continuing to do that in parallel.
Thanks, Ash. So maybe, I won't shy away from the tough questions. So here's another one. I mean, you've been clear. In Canada in particular, we struggle. We don't have even a Teplerson, and I know that's clearly not the most efficient drug, and we're well moving into clinical trials with now, all these new exon skipping. But I mean, what's the possibility for thinking about platform type trials in the exon 51 space? I mean, it seems to me, and I suppose it's the idea with many people here, if there's a drug approved in the US for exon 51, why are we not using that as our standard in clinical, instead of placebo, going ahead in clinical trials of 51 skipping?
Yeah. So I won't, it's a great question. I won't pretend to be a regulatory authority. Maybe I'll offer a couple of thoughts, but this is up for huge debate. So, one, from a regulatory point of view, there is no exon 51 standard of care. So, yes, it exists in the US, but there is no exon 51 skipping drug that's approved in frankly, really in the majority of the world. So while there is a drug on the market in the US that one could consider standard of care, that's not globally recognized, frankly. And so to do a head-to-head study against a drug that's really largely unapproved outside of the US from a regulatory standpoint probably has certain issues. Now, that said, I don't think it moves us away from thinking about eteplirsen and what, kind of how much, dystrophin it produces, the less than 1% dystrophin it produces as a benchmark, as something that not just Dyne, but many other companies are trying to actually do much, much better than what's out there. So it does provide a type of benchmark. Now, I didn't address your question on platform trials. I think this is something that the advocacy community, the physicians, healthcare providers, and companies have been struggling with for some years now. And I do think there's a place for platform trials, but a lot of things have to come together in terms of being able to do a platform trial across products, across companies. And it's complicated because you have so many nuances around inclusion, exclusion criteria, nuances, you have, different designs that companies want to put forth, timing of the development of those programs. So a lot has to come together for platform trials. And so I think there's a lot of discussion that needs to be had. It's of course easier to do it within a single company that's moving forward multiple exons.
Yeah, no, thank you. Okay, any other questions from the audience or online? No, thanks so much for joining us.
Craig, thank you for your help.
Thank you.
Dr. Dugar, a pleasure. Always great to see you, and thank you so much for the update. We appreciate you being part of this.